Thank you, Laura, and thanks everybody for attending the first intern seminars of the season for the Avian Ecology Lab. So I'd like to introduce our first speaker, Skyler Bol. Skyler grew up in Northern Colorado. He and all his family were outdoors folks, and so he spent as much of his time outdoors and really became incredibly proficient at bird identification, but just an all-around really great natural historian, which of course sets you up perfectly for a career in biology. He went to Northern Arizona University in Flagstaff, which again is sort of embedded in the kinds of habitats that I'm sure Skyler would like to explore. He also sought out jobs that sort of mingled his interests. He worked for Knowles National Outdoor Leadership School in Lander, Wyoming, which is in the Wind Rivers. But he also worked in places like in around Flagstaff. His next job is actually with Hawkwatch International, which is a great job, but it's also in the North Cascade. So he's making an effort to mingle those things. At Northern Arizona, he majored in both biology and wildlife management. And while he was in college, he had a few different experiences, but both of which led him down the road toward an interest in disease ecology. He worked on some tick-borne diseases in birds, where he learned a lot of techniques such as capturing and bleeding them. But he also worked in a pathogen and microbiome lab in a level 2 facility, where he really got the deepest experience in trying to understand disease. And that led him to his project now, which is looking at avian parasites and what influences their susceptibility to them and the consequences of having them. So with that, I'll turn it over to Skyler.
Awesome. Thank you, Reed. I think people should be able to hear me pretty clearly. Great. So yeah, I guess I'll just go ahead and start this off. Hi, everyone. My name is Skyler Bol. I'm one of the interns here at Archibald in the Avian Ecology Lab. And I decided to do my research project here on the susceptibility and consequences of blood-borne parasites in Florida scrub jays. So we'll start off here with some just general background. We've known about blood-borne parasites in the jays here for over 50 years. Researchers back in the 1970s found over 15 different species of protozoan parasites in these jays. There's a couple different mechanisms where these parasites can be transmitted to the jays, and then there's also multiple different vectors. So in the classic sense, we could think of this as an issue of like mosquitoes transmitting a disease from host to host, but then there's also some more complex dynamics going on here. So a couple different parasites that we found in these jays can be transmitted through pretty complicated cycles where there's both intermediate hosts and definitive hosts. This creates some pretty complex transmission dynamics, and that's kind of what got me interested in this problem here. So some of the infections that these blood-borne parasites can incur have a pretty wide variety of like effects. Some of these infections can be latent, where there aren't very many symptoms in the host, and then things like avian malaria or malaria that can be contracted by humans have pretty high mortality and can be quite dangerous for the host when they get infected. So the two parasites that we mostly study here at the station in these scrub jays are microfilaria and trympanosomes. We'll go over some general background on these guys just so that you guys can get a kind of baseline knowledge. We'll first start with microfilarids. So this is what they would look like under microscope. And these guys are in the early life cycle of parasitic nematodes, which on their like adult stage are called filarids. These things are commonly known as heartworms and roundworms, which you might have heard of before. And they can be potentially harmful to the hosts that they're infecting. Another issue with microfilaria is that they can get wrapped up in this thing called microfilarial enhancement. This is when they're inside the vectors, and as they progress through their life cycle, they can actually make that vector more competent at spreading other pathogens like bacteria and viruses. Trympanosomes are the other kind of parasite we have here at the station that we find in the scrub jay blood. These guys are a little smaller, and they're also pretty diverse. So we have a couple different species we found, and then they have a pretty low host specificity. So what this means is that The trympanosomes we see in the scrub jay blood could also potentially be in other avian hosts here in the scrub ecosystem. Trympanosomes are most commonly transmitted through flies or biting midges. And a lot of the times, these things are actually ingested. So scrub jays see big, juicy horseflies flying around, and they're going to want to eat that. And what they don't know is that that horsefly might actually be contaminated with these trympanosomes. Great. So on to the questions. For my research project, I was really curious in understanding what factors were going to influence a jay's susceptibility to these blood-borne parasites. And then once infected, I really wanted to understand what kind of consequences these blood-borne parasites were causing. So I expected to see that endogenous factors, things like general health and immune strength, and exogenous factors, so things like the climatic variables and population dynamics, were influencing whether a scrub jay becomes infected with these blood-borne parasites. I also expected to see that the blood-borne parasite infections caused a change in the short-term and long-term behavior of these individuals. So a little bit on the samples that we take. We bleed the jays here when they're in a stage of nutritional independence, where they're kind of going into the adult stage of their life. This is when the birds are no longer dependent on their family groups and parents for food, right around 65 to 85 days into a jay's life. When we bleed these jays, we take that blood back and we separate the red blood cells from the plasma. And then under a microscope, we can actually look in that plasma and count the amount of microfilaria and trympanosomes. With this sample, we can measure too how much red blood cells there is relative to the plasma, and then that's called a hematocrit level. So we store that data as well. All this data that we collect, we keep in our long-term database here at Archibald. And I was able to use data from 2010 to 2021 for the analysis here of my research. For my project, there's two kind of main questions I was asking, so it seemed most logical to kind of put it into two parts of research. First, I was interested in studying susceptibility. So this is going to be how the jays are getting these parasites. And then I also wanted to understand the consequences. So this is going to be the stuff on how these parasites are affecting the scrub jays. We'll start off with susceptibility first here. So we're going to be understanding what factors impact a jay's likelihood of infection. There's a lot of different metrics I tried to assign here just because these complex transmission cycles have a lot of different aspects to them. So yeah, I chose a couple kind of well-rounded metrics here to try and understand that. Group size I used as a measure of host density. So if there's more scrub jays in these families, it's possible that having them being closer together there will increase the likelihood of transmission between individuals. I measured the distance or calculated the distance from nest to water source or seasonal pond nearby, and that's going to be a measure of, looks like we got both, vector density. That's going to be, so we know these seasonal ponds are breeding grounds for things like mosquitoes and biting flies, which are going to be the vectors for these blood-borne parasites. And if jays are hanging out closer to those water sources, it's possible that that's going to increase the amount of vector-host interactions. Annual precipitation I used as a metric of vector abundance. So this is similar to vector density, but here we're just looking at the total amount of water and how that increases the amount of vectors in the ecosystem without any dependency on the density of those vectors. I chose to look at hematocrit level. That was going to be kind of my measure of immune strength and whether these birds were at any kind of whether their immune levels were impacting whether they got these parasites or not. Body condition, I chose to measure as well. That was going to be my metric for general health. So how much nutrition were these birds receiving earlier in life and whether they were getting enough parental care and could develop properly. So for the analysis of my susceptibility portion, I used two different main equations. I took all the variables from the previous slide that I thought would be influencing these things, and I compared them to both infection status and parasite load. So infection status, that's going to be looking at whether a bird is infected with these parasites or not, and I wanted to see whether that was a function of all the different variables. Parasite load, I also wanted to understand, so how many parasites these birds had, and whether that was impacted by the same list of variables. I analyzed my results for this project in R, and I used general linearized models as kind of a framework. For my infection status analysis, I used a logistic regression. That's because infection status is a binary variable, so birds are either going to be infected or not. And then I used a negative binomial regression from my parasite load general linearized model because of overdispersion in the data. So I had a lot of birds that had pretty similar amounts of parasites, and then a couple outliers that had a lot more parasites than the rest. Awesome. So now we're going to kind of hop into the results and see what the susceptibility analysis found. So if we look at this first figure here, we can see that there's a pretty big variation in the amount of infected birds each year, as well as the amount of infected birds for each type of parasite infection. The number N there, that's representing how many birds we're able to sample each year. And one interesting thing to notice about this graph is that the years where we have the most birds infected with microfilariids are not necessarily the same years where we have birds infected with the most trypanosomes. So that kind of suggests that there might be different factors influencing how many infected birds there are each year. Next, I was looking at parasite load. So I wanted to understand the variation in this project timeline of how much parasites were in each bird. You can see here that there's kind of similar variation in years where not all years have the same amount of parasites in each bird, and that there's also different number of parasites for the different kinds of infections. So microfilaria are more often found in high numbers in birds and trypanosomes in low numbers. So now we're going to look at some of the factors that we were talking about earlier. I wanted to understand what factors are impacting a bird's chance of contracting these blood-borne parasites. What I found was that average breeding season precipitation and distance from nest to pond significantly impacted the likelihood of birds getting infected with microfilaria. I also found that average breeding season precipitation significantly impacted whether or not birds got infected with trypanosomes. So we can see here that in years or in infected birds, there are more often, this is for trypanosomes, preface, birds infected with trypanosomes were more often found in years with higher rainfall, and birds that were uninfected were more often found in years with low rainfall. This figure here is looking at wetland distance from nest, and we can see that for microfilariids, birds that were uninfected were slightly further away from wetlands than birds that were infected with microfilariids. Okay, so then for parasite load, the only significant result I found was that average breeding season impacted the number of microfilariids that were in these infected birds. So here on this graph on the right, we can see that there's a general positive trend. So what that means is that in years with higher average breeding season precipitation, there's more microfilariids in birds that have been infected. Great, so what does all this mean? It really shows us the importance of water in this transmission dynamic. So these seasonal ponds that we have here in the scrub, they have a pretty big influence on the likelihood of a bird getting infected, as well as the amount of parasites that that bird is going to contract. It's also important to notice that these two variables that we were measuring, looking at water, were kind of our indicators of vector interactions. So it shows us that vectors are a pretty important part of this transmission cycle. And I think that it'd be really interesting to take this a step further and to look at whether there's more vectors in these areas or whether it's just an issue of these vectors coming into contact with the host more often. Great, so that's the susceptibility part. Next, we're gonna hop into consequences. So we're trying to understand here, are there any short-term or long-term effects of these parasitic infections? For this, there were four main effects that I hope to measure. First, I wanted to understand whether parasites had an impact on the bird's survival. For that, I chose to measure first year survival as a binary variable, whether a bird survived that first year. I also wanted to understand whether parasites impacted the breeding. So for that, I measured it through age at first year of breeding. So kind of when the bird matured to a breeding level, breeding social status. And then I also measured it with the number of fledglings produced in the bird's first 5 years. I wanted to understand kind of the physiological response, whether there was something internal with the bird that we could see as a result of these infections. And I wanted to measure that through looking at the hematocrit levels. The fourth category that I hope to investigate was whether there was a observable difference in the short-term behavior of these birds following these infections. So for that, that's the aspect I decided to use for field work for my project. During my time here at Archibald, I was able to perform these things called focal follows. And that was kind of my take at understanding whether there was individual behavior that was changing as a response to these parasites. So for these focal follows, I would go out into the scrub and I would follow each bird for an hour long, recording what behavior it was doing and where it was doing that behavior. I categorized the behavior in a couple different types, and the birds that I was following were birds from the previous year, so from 2021, because those birds had most recently been sampled for blood parasites, and we most recently understood their infection levels and status. So once I had gone out in the field and recorded all these behaviors and how much time the birds spent doing them, I was able to create a time budget. So understand the frequency at which these behaviors were occurring in these birds' daily lives. From there, I could compare and see whether there was a difference between the uninfected birds and the infected birds' short-term behavior. Okay, so that's a little bit on the focal follows. For the rest of the analysis, I chose to use a t-test and Manu-Whitney tests to understand whether there was that difference between infected and uninfected birds for all the different variables I hope to measure. If there was a significant difference between the groups, I would then look at whether parasite load, so the amount of parasites the birds had, was correlated with these different effects. And for that, I was using regressions and general linearized models again in R to analyze. Great. So now we're going to hop into the results section of my consequence analysis. So for consequences, I was trying to understand, just to reiterate, that we were looking at how jays are impacted by these blood-borne parasites. I found that there was a significant difference in both hematocrit and first-year survival for birds that were infected with microfilaria. And I also found that there was a significant difference in first-year survival for birds that were infected with trypanosomes. Great. So first, we'll look here at the hematocrit results, and we can see that infected birds had higher hematocrit levels than birds that were uninfected with microfilaria. This is kind of to be expected. It's a pretty common occurrence that there's some kind of immune response to these infections, whether benign or not. And hematocrit here is just showing us that the immune response, that the birds that are infected are incurring some kind of immune response to fight off these blood-borne parasites. So first-year survival actually had some pretty interesting results. It wasn't really what I expected to see, but the birds that were infected with both microfilariids and trypanosomes actually had a higher first-year survival than the birds that were uninfected. So kind of an interesting result. So the negative results here, I didn't see any response in short-term behavior or breeding success. So parasite load didn't impact anything with the short-term behavior that I seek to measure or any kind of breeding of these jays. Parasite load as a whole didn't have any effect on the characteristics. So the amount of the parasites that these birds had didn't really impact any of these factors that I measured. Great, so just a little bit about these results. The difference in hematocrit levels, as I kind of just mentioned, show that there is some kind of immune response these jays are having to these parasites. That's to be expected, whether it's really changing the bird's demography or behavior. We didn't really quite see that as a significant result, but it is important to know that there is some kind of physiological response to these infections. So first-year survival, it seemed really odd that there was a higher first-year survival for infected birds. And it's possible that maybe these benign infections aren't actually causing some kind of negative effect, that maybe this is a beneficial kind of prepping for other more harmful pathogens that the bird might encounter later on in its life. It's also possible that there's some kind of other factors or other kind of confounding variables going on here that we didn't quite measure to the best of our ability with these metrics. Short-term behavior, there wasn't any response, which it could have been possible that we would have seen something if the birds were infected last year, but a more accurate analysis would be to do these focal follows and try and understand the short-term behavior during the infection. So this would be best done by watching these birds immediately after they've been bled, or maybe within a week or so that we could understand a change in behavior during the infection. Great. So yeah, that's kind of the majority of the research I've been able to do here at Archibald. I think there's a lot of really cool directions that this research could go. Understanding these vectors further and kind of filling in the gaps here with this puzzle of how these different blood-borne parasites are being transmitted and how these vectors are interacting with both the host and the parasite could really kind of elucidate this relationship. And then I also think that determining all the competent avian hosts would be really beneficial here. So it's possible that these issues affecting the scrub jays are also driven by larger ecosystem processes, and there could be other potential hosts that are impacting um, the prevalence and susceptibility of these parasites within the jays themselves. Awesome. Yeah, guys, that's my presentation, and I would really just like to give a shout out to some folks here. Archibald is such a great place, such an amazing community. Everyone here in the Avian Ecology Lab is like a family. They've been so supportive and have taught me so much throughout the season. Reed especially, I mean, a really great mentor. I really want to thank Fabi, Liz, and Greg as well. They've been super helpful. They got us out working in the field with them and seeing other biologists like enact actual powerful conservation is what gets me fired up. It's what gets me excited to keep pursuing this career. And then, yeah, I also wanted to thank Kevin and Julie. They really helped getting access to some of this data for this project. And then shout out to all the Archibald interns and RAs. These guys are the best folks out there. I mean, these guys like even more like a family. They're yeah, a wonderful group of people, and I'm really sad that I'm gonna have to say goodbye to them in a couple of days. But thank you everybody for tuning in, and yeah, that's is about as much as I have for you. We'll get into some questions here.
All right, Tyler, Skyler, there's a bunch of questions in the Q&A, so I'll just start with them.
Great.
Pam Moran asks, can infected birds' immune systems clear the parasite loads, or is a bird infected for life?
Yeah, so that's a good question. It's a really kind of interesting transmission cycle. So with some of these parasites, the birds can actually have an infection and not show any microfilaria. So these parasitic nematodes, when they're in their adult stage, they actually can be inside the host, and we won't understand that unless we see them producing microfilaria. So those will cycle back through the birds. A lot of the times, it's just one part of the birds or one part of the blood parasites transmission and life cycle. So eventually, yes, birds will be recovered and they can actually get infected multiple times, to my understanding.
Great. Aaron David asks, is there any seasonality to infection by either pathogen?
So that wasn't something investigated yet. I'm actually collaborating with some other researchers here on this project, looking at differences between wet season and dry season and how that impacts this disease transmission cycle. I would assume that this is a process that's pretty heavily driven by water and seasonality here, just due to the abundance of vectors in the ecosystem and the likelihood of transmission.
Great. Meredith Howell asks, if you could return to participate in the same study next year, how would you further the study? What changes would you make?
Oh man, I think that there would be a lot of really powerful analysis in kind of lab techniques, going in and taking maybe blood samples from vectors and understanding from there how they're being able to like look at sequence data and understand who the vectors are biting, kind of the rates of transmission and things like that could be really useful for this understanding this whole system.
Good. Ella Siegel says, great job, Skyler. Do you know if there are any vectors that transmit parasites to jays that don't tend to live near water?
Ooh, you know, that's a good question. I mean, I know from personal experience that horseflies are hiding like under every leaf in the scrub right now. So that could definitely be something where they're spreading out and not certainly clustered around water. I know that there's some of these parasites that are they are transmitted through frogs and ranids. So I think that there's a lot of different kind of dispersal of those guys too. I definitely think that there's some vectors that do not depend on water.
I would add to that we've done other work with other diseases too. And for example, no-see-ums transmit a fairly severe disease, but they are, in that case, it's elevational. So they're up higher, and blue jays roosting in trees are susceptible to them, but scrub jays roosting in shrubs aren't. So, and they don't have anything to do with water. So, Jack asks, what would be the benefits to the birds of living closer to a body of water?
Ooh, that's a really good question. There's a lot of really cool research, I think, that Reed and collaborators are about to start on with that. I know that there's some research looking into different kinds of proteins that might be a source of kind of nutrients for these birds in these wetland ponds. So I think it could be a really interesting aspect to kind of look at that hand in hand with this disease transmission cycle and understand. I mean, it's definitely kind of probably a cost and benefit thing. So you might be at a higher risk for these pathogens, but you also might be getting some kind of nutritional support from the wetlands that you can't find in other places in the ecosystem.
Someone else asked, do the jays transmit these parasites to their predators or other species? Or are either of these parasites lethal to the jays?
I don't think either. So both microfilarids and trypanosomes haven't been documented to be lethal. We saw that there's that kind of first year survival correlation there that looked like it was actually higher for infected birds. I'm not sure about upcycling, whether birds that are feeding on the jays are going to be more likely to get them. I would assume that there's a certain level of host specificity with these blood parasites. This is kind of speculation, but I would assume that there's a big enough gap between maybe predatory birds and jays that the same kind of parasites won't impact the two different groups.
And I apologize to Christine, I jumped over you. Are there any parasitic co-infections where certain infections tend to show up in the same birds?
Yeah, so I
Yeah,
actually did some research here looking at co-infection of these two different parasites. I created a little mathematical equation to understand the frequency of each parasite and whether they're more likely to be found co-infecting or in a solitary infection. And I saw that there was a little bit of preference in both parasite to solitary infection as opposed to any kind of co-infection. So.
Skyler, we have a couple more questions that we'll save. Maybe you can address them online afterwards, but we want to proceed on to our next seminar. So thank you again, everybody. Great questions and great seminar, Skyler.
Thank you, guys. Okay.
So I guess it's up to me to introduce our next speaker. And Brian, if you could share your screen so that we get your title page up, that would be great. Our next speaker is Brian Cammarano, who is a Florida native, born and raised in Miami, used to our heat, so we made him do all the work. Brian actually has a bachelor's degree in business management that he got back in 2015. And then he wised up and got a bachelor's degree in ecology, evolution, and conservation science from University of Central Florida in 2020. Brian is a really avid birder, as was Skyler. And Meredith had actually met Brian several times chasing rare birds around the state prior to his application. And, you know, one of the things we always talk about is how do you demonstrate interest in a letter and a CV? Well, having met the people doing what we want you to be doing for your application is probably no better measure of interest. So when Brian applied, we were immediately interested in stuff. Immediately after getting his bachelor's degree in business, Brian really started an aggressive campaign to get lots and lots of jobs in biology. And they took him all over the place to Africa. He worked in Biscayne Bay, where he worked on white-crowned pigeons. He worked in Montana. And he got lots and lots of different experience. He more recently has worked with shorebirds with folks down at Florida Gulf Coast University. So I really should have said this during both of their presentations, but Brian and Skyler were truly birding buddies. And I've never had interns that were out doing just bird watching on their spare time, you know, a busman's holiday, out all the time. And it was really amazing because a lot of times on weekends, I'd see a post on Facebook of the newest life bird that they found that morning, usually before I had my coffee. So they were great, and it spilled over to the lab. We had a lot of great lab birding trips. So Brian is going to present his work on I just drew a total blank, Brian. You tell the title.
All right.
So with that, I'll turn it over to Brian.
Perfect. All right. Thanks, Reed, for the introduction. Just give me 1 second. All right. So my name is Brian. I am an intern here at the Avian Ecology Lab. And the title of my project is Sorry, Please Try Again. And I investigated the change in nest site characteristics following a failed nest attempt in the Florida scrub jay. So we'll start out with just understanding the fundamental ecology of the Florida scrub jay. They are very habitat specific. That means they require relatively low open growing scrub habitat dominated by oaks. They create these family groups, and with those family groups, they maintain permanent territories. And although they're cooperative breeders, meaning the helper birds within the family help care for young, the breeding pair are the birds that build the nest without the assistance of helpers. So the nesting season begins as early as late February and continues into June. So that's their window there to attempt to hopefully successfully reproduce. And now we'll look at the nesting requirements that they have. The majority of the oaks that they nest in are Quercus geminata and Quercus mortifolia, abundant in the scrub. There's also a few other common oaks in the scrub that they utilize, Quercus chapmani and Quercus inopina. And then with that, there's always situations where they'll nest in other similar shrubs in the scrub, and there's just a handful of those. So this nest, it consists of a platform, and that platform is built from twigs. And within that platform, we have a nest cup, and that nest cup's lined with palmetto fibers. So now we'll talk about the pressure that these predators put on the jays during the nesting season. We have about a 40% chance of nest success, and the primary cause being predation for nest failures. And so once a nest is predated, they'll have to build a new nest. So they'll move on within their territory to a new spot and build a new nest. So if they are successful, though, they'll rarely attempt to nest again within the same season. Right here, you can see that 67% of egg loss is due to predation, and 85% of nesting loss is due to predation as well. So you can see how much of a pressure predators put on these jays. Now we'll take a look at the nest predators. The nest predation is attributed, most of the nest predation is attributable to diurnal snakes and birds, the high majority being snakes. Nest predation significantly increases throughout the nesting season. So a nest in, let's say, March is much more likely to fledge young, meaning a successful nest, than a nest in, let's say, June. Snakes will pretty much come by and consume all the contents within the nest and leave the nest empty. So there you can kind of investigate when you're nest checking to see what kind of predator came across. A bird would most likely leave some kind of evidence like a cracked egg. Scary snakes. So with that, that's my background. We can think about the questions that I incorporated into my project. So the first one being, do nest site characteristics differ among first nests that are successful, depredated, or abandoned? So there we're looking at all the first attempts and their fates and whether there's differences in nest site characteristics between them. Next question, do nest site characteristics differ among nest attempts within a season, regardless of nest fate? So just as the season progresses, is there any kind of trend in attempt number? Are there changes between those? And then third question, we have, do nest site characteristics in replacement nests differ relative to the fate of the previous nest or nest attempt? So meaning these nests fail, they're either depredated or abandoned. What kind of, what difference between those two are we seeing? in the subsequent nest. I'm also interested in comparing the subsequent nest attempts between those that were successful and those that failed within a year. The only problem is, like we mentioned before, pairs that are successful don't build subsequent nests. So in order to test that, I thought of a question, which was, do nest site characteristics differ between a pair's previous year's successful nest, meaning last year's, and its first nest the next year, with pairs that fail from that first attempt and their subsequent nest. So looking at the changes between those two. So here I have my hypothesis, very general: reducing the risk of predation drives nest site selection. So from that hypothesis, I can draw some predictions. Nest site characteristics should differ between nests that are successful or failed. So from the fate that they have, what differences are we seeing there? I predict that successful nests are likely higher and more concealed than nests that fail. Because nest success declines throughout the season, nest site characteristics should also vary relative to attempts. So there I predict that later attempts in the season should be taller and more concealed. I also predict that since we know that nests fail from predation or abandonment, subsequent nests of those that failed from predation should be taller and more concealed. So we should see a lot more change in those than those that fail from abandonment. And last, I have pairs successful in the previous year will select sites in subsequent years that differ less. So since they were successful last year, we'd expect less change than the subsequent attempt following predation within a year. So from all that, I set out to the field to collect my data. Field measurements included GPS coordinates. So there I can look at the distances between nest attempts. We have nest height and shrub height, measured those. And from nest height and shrub height, we can measure relative nest height. Relative nest height can be thought of as if we have a 1 meter tall nest and a 2 meter tall shrub, the nest is in that shrub, we have a relative nest height of 0.5. We're also going to identify the shrub species, and I incorporated this photo of cover. So in order to measure nest concealment, I took a photo from the center of the nest cup up and quantified that. So I'll go into more detail with that in the next slide. And all this data was inputted into field maps. So looking closer into the calculating nest concealment percentage, I utilized a software called ImageJ to quantify nest concealment percentages. So there on the left, you can see the photos that got put through ImageJ. I was able to put them into black and white and then just quantify the black space. And that was a standardized way of seeing how much cover each nest had from above the nest. From all that data collection, I was able to summarize the nesting season for 2022 here at Archbold. We had a total of about 104, or we did have 145 nests. About 22% of those were abandoned. About half of them, so 50%, failed, and about 27% of them fledged. Shrub choice was pretty consistent with the biology. We had about 43% in Quercus geminata, about 31% in Myrtifolia, and the other 26% was just a variety of other shrubs. Some notable means here, we have nest height. The average mean between all nest attempts was 1.15 meters, shrub height 2.22 meters, relative height being consistent with those averages of 0.53, distance from previous nests about 200, an average of 200 meters between each attempt, and nest concealment was a very strong 81% average. So before we begin this, I'd like to mention that I ran all my variables and analyzed them all, and the only variable that should prove to be significant was nest height. So for the remainder of this presentation, we're going to focus on nest height and the changes in nest height. So first off, we have comparison of nest height between first attempts. So very general, we're looking at just the first attempt of the season and its fate. So compared to its fate. So we have abandoned, depredated, and fledged meaning successful. From the first attempts, we found no significant differences in nest height or concealment. There's pretty similar distributions among all fates. So what this helps suggest is that nest height does not influence the likelihood of nest predation or abandonment. Next, I analyzed nest height between attempts within a single season. So here we're just looking at attempt number throughout the season and what changes we might see there. Nest height, there was a trend of increasing nest height as attempt number increased. It's important to note there, if you see the letters A, B, and B, A was significant to B and vice versa, but AB was not significant to A or B. And thinking about alternative hypotheses here, we used the Acorn database and measured the 2020 to 2021 oak growth over a year and compared nest changes between successive nests. And we found that the average oak growth was about 0.05 meters. And we compare that to the 0.24 meters that just a few months had a nest increase. So this helps kind of explain why we can disregard plant growth as being the reason why these nest height increase. Next, I looked at the change in nest height due to previous nest fate, particularly looking at first attempt and second attempt. That sample size was large enough to really get a good grasp of what was going on. And we found that successive nests following predation were higher than successful nests following abandonment. The change in nest height between first and second attempts increased by an average of about 0.25 meters when the previous attempt was depredated. And so when I ran the same thing with attempt 2 to 3 and ran the comparisons, I did find a really high p-value there. And that could be in part due to the small sample size or a handful of other factors. And sample sizes between 3 and 4 and 4 and 5 were just not even big enough to analyze. Next, we think about that successful nest from the previous year. We have the two groups. In order to compare change from a successful nest, we had to use that previous year. So we have successful nest in 2021 compared with first nest in 2022. And then the next group would be the first nest failed in 2022 from all those first attempts, the ones that failed, compared with subsequent nests in 2022. So the changes in there. And we found that nest height differed less after a previous successful attempt than did with a nest predation. So kind of what we expected. And from that, we can draw some conclusions. Again, I wanted to mention that the only variable tested for change in response to a predation event that proved significant was nest height. So out of all the variables like shrub height, relative height, nest concealment, only one that was significant was nest height. Nest height increases as attempt number increases. So regardless of previous nest height or nest fate. And we can also say that that was not a result of natural plant growth. Previous nest fate influenced the change in nest height of successive attempts between first and second attempt. So whether they all failed, but whether it was abandoned or depredated, we saw a lot bigger change in the depredated nests. And then information gained by previous year's positive experience has an impact on the following year's first attempt. So the analysis showed that the successful nest last year kind of provided some positive information that passed on to the next year, and they changed less in their next attempt. So from all that, we can think about the future study of this. We'd really want to kind of compare across multiple years, not just one season. This will kind of help any kind of year bias. All these measurements that I took, most of them are collected on an annual basis throughout every season. So we do have that historical data. So it'd be great to utilize that historical data. That'd create much larger sample sizes, and we'd be able to test for consistent significance among all these years. You could also incorporate a behavioral component. We know that family size and nest defense help reduce nest predation. So incorporating that could possibly lead to new findings. Also, a few more variables like habitat structure is always good. Side concealment, visibility, nest location in relation to open areas. So those last two could possibly lead to earlier predator detection if it's an open area. and that can also contribute to maybe a reduced predation, nest predation. And then a completely different approach to this project could be looking at habitat availability versus breeding pair preference. So seeing what's available in their territory and comparing it to the preference of that breeding pair. So with that, just want to say thank you to everyone, especially Dr. Reed Bowman. He was a great mentor, guided me to success, and we'll see where I go next. But then we have Meredith Heather. She's a great supervisor. She is a role model to me. Bird Lab and Avon Park crew. Bird Lab, just really hardworking team. We did great this season. Avon Park crew, so many opportunities there to help out with research, learn new skills. Skyler Bol, shout out to him. He's the R god. He taught me a lot of the basics of an R, and just any question that I'd come up with while we're in the room. He's my roommate. He would be more than willing to help. Shout out to Fabi. She helped me create my field maps for my project. Julie, every GIS question I had, she was able to help me with, and she was more than happy to help every single time. Everyone that lives in the lodge, just coming back from the field every day and just seeing what's going on during lunch and dinner time is just really good to decompress after being in the hot scrub all day. And of course, all of Archbold's full-time staff for just keeping things running smoothly here at Archbold. And with that, thank you to everyone listening and attending. And does anyone have any questions?
Great job, Brian. There are a couple questions. Pam asks, are FSJs more highly depredated than other bird species? And is there anything that can be done to reduce FSJ predation?
Well, what I like to say with that is there's not really much we can do about what goes on in the scrub when it comes to predator-prey interactions. It's just the natural flow of things. I'm not sure about other birds. I'm sure other birds have just as high as predation, since these snakes are finding nests through olfaction. So yeah, I don't think that maybe that's not the focus of how we can really like incorporate conservation into the jays. I think habitat structure and maintaining land management plans would be a lot more useful.
Okay. Bill Hawthorne says, great job. Sorry if I missed it, but was there a difference in nest height after predation from a snake versus a bird? I wonder if they move up after a snake predation and down after a bird predation.
Yeah, so there was just so few instances where when we checked nests, there was evidence of a avian predator. I can only remember one during the season, during my checks. It's just the percentage of snake predation is just so much higher than anything else that we would kind of see the trend of what the influence of snake and terrestrial predators has rather than avian predators.
Kelly asks, do you think breeding pair duration influences nest site selection and nest characteristics?
Yes, possibly. That'd be something to explore for sure, because I didn't look at how many times these birds have successfully bred. So yeah, experience. If I found that information was gained from last season, then there could definitely be opportunities to explore that to see how much better, I guess, they get at creating their nests.
And Emily asks, what are the causes of nest abandonment?
That's hard to say. There's just some birds that abandon for whatever reason. It's just a handful of, I can't think of any on the top of my head. But yeah, it's hard to get down to why that bird or that pair abandoned their nest. It's not as easy to say, compare it to predation. So like if contents are missing, that nest was predated. Yeah, abandonment is a bit more complex, I think.
Fantastic, Brian. That's it on the questions. Let me just check the chats. The chats just say a whole bunch of wonderful job, great job, excellent job. And maybe there's one last question. Yeah, there is one last question. But
Okay.
this is a question for Skyler, so we'll address it later on.
Okay. Perfect.
Okay, well,
I'm gonna
thank you,
stop sharing.
Brian. And I just again want to thank everybody for attending these, and make sure to come back in mid-September for our next round of avian ecology intern demonstrations.
Wow, thanks you guys. That was a great job from both of you. And thanks again to everyone for attending today. Stay tuned to our website and newsletters for upcoming events in August and September.